passar à frente da nossa prova de quebra.
I just saw that like this, I'll do when I give a presentation, so I'll do suggest something.
Reviewing order.
I just realized that now actually, thinking about it. I say more than what I can do. I will use and I don't know what
The way that I don't know, Chris is coming. So, yeah, I will introduce you and you look like the. So I will start now. So good afternoon, everyone. So thank you for attending our seminar. It's a great pleasure, and everyone on YouTube and online as well. It's a great pleasure to introduce you Professor Alain Goossens from VIB. He's here as part of a collaborative project with Magda, and we are honored to have Alain here to give a talk in our course. So this is a graduate course on biochemistry, molecular biology of plants. And we all have like different, every year we invite several different researchers to present the state of the art of their research. So I'm very happy to have Alain here. So we have 45 minutes more or less talk. And then we open for questions. Questions can come from here, the audience, of course, and the students that are online, and also people watching on YouTube. Okay. So, and then on behalf of the department, the institute, and the responsible for the course, we thank you for your contribution to our course, and the word is yours.
Okay, thank you. Good afternoon, everybody. Boa tarde and buenas tardes to everybody who's online as well. First of all, thanks again for the fantastic 3-4 days that we had this week. And also Nikos, my student, enjoyed it very much. As you will see from my talk, I work on bioactive metabolites. And both for me and Nikos, it was fantastic to see that all the plants that we try to make grow in our greenhouses or in Belgium are just 10 times bigger here and probably produce much more than we will ever produce in Belgium. So this is an amazing environment for somebody, for people like us. Okay, so I'm from the VIB and the Ghent University in Belgium, and my talk will actually reflect the philosophy of what we do over there. So we're very much encouraged to do basic fundamental research. We trying to find new things in plant science in this case. But at the same time, we have to try to look for a topic that also has applications. In this case, is the production of bioactive metabolites that serve all kinds of human purposes, and that will become clear from my talk. So, small introduction to plant metabolites. So, I just took here the example of a root. So, every eukaryotic or living organism produces so-called primary metabolites, which are there to build the organism. Lipids, nucleotides, amino acids, sugars. Every organism needs them and needs them to grow and to build their cells and to maintain integrity. Now, plants, also fungi, many bacteria, have a huge set of additional metabolites, which were originally called secondary metabolites because people didn't know what they were used for the plants. And now they're called specialized metabolites because now we know that they're very important, not so much to make the plant grow, but mostly to actually make the plant thrive and survive well in its environment. So you have lots of compounds which are there to communicate with other organisms, to defend the plant from other organisms, or to actually enter in symbiosis with other organisms. There are also lots of metabolites that make the plant go with the environment in terms of abiotic elements, UV, heat, drought, whatever. Every plant species has a specific mix of metabolites which are really essential and actually as essential as the primary metabolites to make it survive. Now, why are these specialized metabolites so interesting? Well, many of the drugs and products we use are actually derived from these plant metabolites. So, for instance, here you have artemisinin. It's actually one of the last active compounds against the plasmodium parasite that causes malaria. We have anti-cancer drugs. We have painkillers, aspirin, originally from plants as well. And then you have also metabolites that contribute to taste and flavor, and also building substances like isoprene, polymers for rubber, for instance, natural rubber. Now, the difference between primary and specialized metabolism is that typically primary metabolism always needs to be there because you always need cells, you also always need growth, so they're always there. That's not the case with specialized metabolites. They're really only made either in specific cells, specific organs, or only in specific conditions. Because otherwise, if you make them and they have no use, it's a waste of energy and building blocks. So I can illustrate this with tomato. So tomato has different developmental stages. You start from the small green fruit and you go up to the mature red fruit. In the green fruit stage, actually it still needs to grow and build, so it has lots of primary metabolism going on. But also it needs to be protected from the environment because the embryo is not mature yet and it doesn't want to be eaten, for instance, tomato. So at this stage, the green tomato will make lots of bitter glycoalkaloids, which will not attract herbivores. At the later stages, the red stage, originally tomatoes had to be eaten, the seeds disturbed by the animals to be to make them spread, and that's why they actually then reprogram their metabolism, and now they become more tasty and more attractive to herbivores, in this case against. That happens by, for instance, reconversion of the bitter alkaloids to a sweeter version, which is called the aspidosides. You also have carotenoids, sugars, and phenolics, and that together makes a nice aroma that also humans appreciate. Now, this looks like a pretty fixed program, but it actually isn't. Stress can happen anytime. There can be fungi around, can be herbivores coming over, and then the program can be reprogrammed. In this case, the network will be reprogrammed by a hormone called Jasmonate, for instance, and Jasmonate will reprogram primary and central metabolism. So we'll do a different type of sugar fixation or carbon fixation, for instance. And again, it will stimulate the bitter glycoalkaloids because it's not good to be tasty at this stage. So basically, the original developed program can be stopped forever or temporarily. Same time, there's lots of additional things that can happen. It can also, the fruit can start, and the leaves can start making volatiles, and this can in turn attract other organisms like parasitic wasps, which in this case literally attack the caterpillars and also try to help the plants to extinguish their predators. Now, if you understand all of this, and this is where application comes into the picture, then if you find the elements that drive all this metabolic network program, you can also engineer it. So if you find the right switch at the right time, you can actually modulate, for instance, tomato fruit development towards a more tasty or more resistant tomato, whatever. Now, as said, Jasmonate is crucial, and you'll see also in the coming slides, the solution is not to spray your tomato forever with Jasmonate, because there is something which is called the growth defense trade-off. So if you would apply Jasmonate all the time, you will have lots of metabolites in the tomato, but they will also remain tiny because the plants will no longer invest in growth and development, and it will just be arrested wherever. So just spraying, not the solution. And that's why you need to understand it and find the right switch. I'm more or less working 20 years on this topic now, and early on I decided to focus on Jasmonate, and I'm happy I did. I hope to be able to explain it in the next slide. So what's interesting about Jasmonate is that it's a very conserved phytohormone. So not really the exact one that you see here, but similar versions of Jasmonate exist from the bryophytes all the way up to the higher plants, if you want to call them like that. And what's interesting is that the receptor is exactly the same. So the receptor system, is super conserved. So it has the same CoA1 efflux protein. I will explain the function later. and the JAS repressor proteins. When you have the jasmonate there, these two come together and they will launch the jasmonate signaling cascade, in this case towards production of bioactive metabolites. And the second interesting thing is that every plant species developed a specific blend of metabolites of which the production is controlled by JA. If you go to tobacco, best way to have more nicotine in the environment, sprays a bit with JA, 24 hours later, you have a super cigarette. Artemisinin, Advarada, the same thing. Tomato, tomatin, the same thing. Our national pride in terms of crops, chicory. If you want to plant janet and make it even more bitter, kids would not like it, but best way to treat again 24 hours with janet. Anti-cancer drugs, especially for this crowd here. I checked this morning sugarcane. I'm not sure if it will affect the taste of the juice you would make, but sugarcane is also responsive to JA, and it will make more polyphenol conjugates, which will protect it more from fungi in this case. Okay, now I mentioned this growth defense trade-off, and what's common to all these plants, if you will apply JA, you will have more of these compounds, but you will also have stunted plants. So this is, for instance, our Roxas model system, wild type, and mock control. This is what happens if you apply 24 hours of JA. It just doesn't grow. You can also see it's totally red because it's full of anthocyanins, but they don't grow anymore. If you have a mutant in the receptor, you can see it doesn't become small, so that the growth is not arrested, so you can see it works, but you see they're also as green, so basically they also don't make specialized metabolites anymore. So showing that if you would start engineering or to knock out the receptor, you will lose the whole system and you will not uncouple the growth defense. So if you want to uncouple the growth defense, which is our holy grail for engineering, you need to find something between the reception and the feedback. Okay, so that's where I started thinking about. And an interesting feature of the jasmonate signaling cascade is that it's almost entirely regulated on a transcriptional level. So meaning that this is the type of effects that are triggered by after the perception of jasmonate. So normally if a gene is off, the gene is in a condensed, present in a piece of gene which is condensed chromatin. When it needs to be activated, the chromatin around the gene opens, then specific transcription factors can bind to the promoter elements, the genes that you want to activate. And these specific transcription factors will recruit the mediator complex, which in turn will recruit the RNA polymerase, and then you will have a fragment of target gene, in this case, a jasmonate stimulated gene encoding an enzyme making bioactive metabolites. And in case of jasmonate, the one that we want is MYC2, and MYC2 does all of this, but it's not as simple as that is jasmonate signaling. So remember, the function of jasmonate is to regulate this growth defense all the time. So this is a situation where there is no stress. So you have MYC2 bound by a repressor complex consisting of jasmin and topus. When there is JA, for instance, when you have caterpillars eating, what happens is that they destroy the membranes, they release lipids, and the lipids are the precursors for jasmonate. But then you get your bioactive molecule, your hormone accumulated, that will bind CoA1 and JAS. CoA1 is an E3 glucan ligase, which means it will post-translationally modify this protein, and then it will be recognized by the proteasome and degraded. You get free MYC2. MYC2 can now recruit the mediator complex, the RNA polymerase, and you get the JA response. Very simple. Same time, the plant has to make sure that it doesn't get stuck in this defense system. Because otherwise, they will stay small and will never be arrested anymore. So at the same time, you have, depending on the situation, you can have different consequences. So MYC2 will activate its own expression, will activate JA biosynthesis. So as long as there's stress, you have more and more JA response and more stress and more defense. However, if the caterpillar, for instance, goes away, because caterpillars tend to taste here and there, so with the early tasting, you will have some mild JA response, but if the caterpillar decides this is not a nice leaf, whatever, it will go away, and then immediately the feedback system, which is activated by MYC2 itself, will be activated, or they will be installed. So the feedback system consists of MYC actually also driving the expression of the JAS repressor proteins. So from the moment your caterpillar goes away, there will be no more wounding, no more JA, and the JAS will immediately stabilize, and you get back to the normal system, and this happens in minutes. So the plant can very easily switch between these two defense systems, and that's all driven by transcription regulation. Now you see here lots of interactions, proteins with metabolites, proteins with DNA, and proteins with other proteins. So the past 20 years, to get these insights, we applied lots of different omic platforms to study protein-protein interactions, to study interactions of transcription factors with DNA, transcription factors among them, and also to study interaction between metabolites and proteins, in this case both bioactive metabolites and plasmate proteins in the cell. And today I decided not to talk too much about our latest advancements. There will be some, but I wanted to show you how we actually got to the point, because in the past workshop I learned that some people here are very scared of proteomics or interactomics, so I'll try to convince them that it's actually not pointing to anybody here. I will show that actually if you start from a basic knowledge and you do it stepwise, it's actually fantastic technology. So I will talk today about tandem affinity purification and RNA-seq, both bulk transcriptomics and single-cell transcriptomics. So I will start with 3 stories on how we get basic knowledge on JVs again, and then I will start with I will finish with one story, how we apply that to the medicinal plant, and then improve the production of the bioactive metabolites from that plant. So I will start with transcriptomics. You will see here microarray analysis. It's a bit outdated, but that's what we had 15 years ago, and it actually served its purpose well. So we started from the knowledge around 2005. It was not public then, but we knew there were the just repressive proteins, that they could bind to MYC, and that they somehow could also interact with CoI1, and that led to the duration of MYC amplification. But we didn't know much more. So you've seen the previous pictures. There were many more proteins there. This is what we knew in 2005. And what we also knew was that there was a complex network of transcription factors acting downstream of MYC2 and the CoI1 reception. Well, we didn't know how exactly they interact and what their role was, and basically people were mostly focusing on long-term stress responses and not so much on the fast metabolite reduction response. So we decided to focus on the fast responses and using a transcriptomics technology, which were actually not used that much at those days. So Lawrence Paulo was one of my first PhD students. He decided to start working with adoptive cell cultures. So another advantage for Jasmonate is that you can basically apply it to every organ of the plant. You can take cell cultures, you can take roots, you can take leaves. They will always respond. Of course, they will make different metabolites depending on the organ, but they will always respond. So whatever system you use, it's okay to find regulators. Lawrence, this is how he looks now. This is how he looked when I was working. I didn't find the picture of myself anymore. I look more or less similar those days. There were only guys with long hair, Michael. So what Lawrence did was microarray analysis. And what you can see here, so he took very early time points, and the ones that are most relevant here is 30 minutes and 2 hours. And this reflects very well the JA cascade. So you see a lot of genes being activated, but nothing being repressed. And that fits with being the first layer of response. It's a transcriptional activator. So we focused on these ones first, and then I'll also briefly address those later ones at 6 hours. So with the early ones, what you do is you actually see all the feedback and amplification loops. So within half an hour and 2 hours, see very nice activation, of literally every single enzyme according to the You also see all the known transcription factors that are part of the J response in this early response. You have the activators, MYC2, you have 1, and they were all known. But you also see the feedback loop. Also the JASs are here exactly at the same time, half an hour. So everything is there to switch it on and off again if needed. What we also found, and this is again a software tool that maybe not exists anymore, MapMan was very useful in its time. With MapMan, you can actually, especially if you want to look for metabolism, you can map all of your activated and repressed genes into so-called bins, which then reflect the different metabolic pathways. And it was very nice here. We saw a very nice activation of the phenolic pathway and also of the amino acid pathway that needs to feed the phenolic pathway. Google will certainly recognize these things. Yeah. So in more detail, what was activated was actually monolignal biosynthesis. So you literally see, again, every single gene involved in the pathway being activated by JA at the transcriptional level, and it's also reflected at the metabolic level. So we see very nice mono and tetralignals being activated or accumulating in your cell culture. So showing that you can indeed work with these kind of model systems and then find the genes if you miss some enzyme in the pathway, and also find potentially the regulators. I will come back to that later. What we also saw with the magma is that actually you have an arrest in the G1, in the G2 phase, and that was nicely corresponding with the phenotype again. So we see a very nice after 6 hours repression of these genes, and that was actually very nice reflected also in both with the FOX analysis. You can see the rest, and also when you just go to cells on medial JA, you see it no longer grow, and actually it means that JA is really targeting within the program the event of a cell cycle. We tried hard to find the link between the regulator and the cell cycle, but so far without success. But again, showing that you can unravel both the growth reversion as the defense activation with this transcriptomics analysis. If you then assemble data, you get models like this, where you have the activation of the pathway through the repression of the degradation of the JAS proteins, and which liberates V2, which then amplifies the cell amplification loop. Specific repressors and activators, including the JAS, that will amplify the signal by making more jasmonate, and that will then lead to growth arrest directly on the cell cycle, and depending on the species and the organ, a certain type of specialized metabolites. So this started to increase our view on the model, but what we didn't know yet was how actually JAS is repressing V2, because JAS is a very small protein, so it was not clear. So for that, we were lucky that here in our department developed a technology for protein-protein interference, which is called tandem affinity purification. And tandem affinity purification can again be done in any type of plant species and organ, as long as you have a genome to capture peptide signals. So here we state our cell suspension cultures, and then we express the tagged protein of interest. In this case, for instance, the JAS proteins. We fuse that to a double affinity tag, and you do a two-step affinity purification, and that allows you to end up with a very pure complex, including your protein of your beta of interest. When you have this purified complex, you do trypsin digestion, and you go to a mass analysis, and you can identify which proteins are there and interacting directly or indirectly with your protein of interest. Now, this is what we knew. So what we did is to tag every single protein that we knew from the receptor cascade or the receptor and signaling cascade, express those in the aerobiotic cells, and then treat those cells either with MOX solutions or with JA, and then see if you have dynamic reassembly of your complexes and which proteins are actually there or not there. And I will go walk through. We did just MIC and CoA1. So we then also do the mock treatment, the JA treatment, and you can see that we had to optimize the system. For JA, we had to work as fast as possible, and the fastest possible was 1 minute. But in 1 minute, everything happens already. So in this case, with the beta JAS1, what we find is other JASs, which makes sense because they can homo and heterodimerize. We obviously find MIC2, and in this case, MIC3 alone, the homolog. We find CoA1, but only after JA treatment, which makes sense because before the JAS would be stable. When you do CoA1, you see the opposite picture. Basically, there was nothing there. Only when you apply JA, after 1 minute, you get specific interaction with JAS12, and then just CoA1 is the 8, nothing else. With MIC2, you see kind of the opposite picture. You have more conditions. MIC2 is bound by lots of different JASs, really to block sensitivity, and it's also homodimerizes, so you also find those ones. And if you apply JA, now you have to apply a bit longer because you really want to get rid of all the JASs, so 1 minute is not enough. You really need a 1 hour treatment. Also taking in mind, remembering MIC2 will also activate the expression of the JAS, so we need to force the system. And then there was only one protein that was there, so big difference to BIB, for instance. So we didn't have much choice on what to work. That was Ninja. Ninja stands for, was nominated by Lawrence for novel interactive or Jas proteins. And we also started working with Roberto Solano those days. And what you can see from Ninja is that it's always there with the Jas, never with CoI1, and only there with MYC2 if you add Jasmonate. So if you want to find out what its potential role is, the first thing you have to do when you do after affinity purification is to find out with which of these proteins it's directly interacting. From this, you can guess it's probably Jas, but still you have to confirm that. So the most used method, still the easiest one, is these two hybrids. I'm not going to expand on the technology here, but in this case, you have you fuse your two proteins of interest to the two different halves of a transcription factor that's active in yeast. If you have interaction, your yeast will start making an amino acid that is lacking, so you can grow your yeast on an amino acid deficient media. If you do that, for instance, between MYC and the Jases, you can see that MYC2 is, all the MYCs are bound by literally almost all the Jases, and this is reflected on this. A transcription factor related to MYC that cannot bind Jas will not grow on the media. Now, if you do that with Ninja, we could prove that it was not interacting with MYC2, not with CoI1, but basically with almost all the Jases. Another advantage of these two hybrids is that you can actually start looking into your protein and see which domains are there. So in this case, Ninja has 3 potential domains, and Jas was known to have 2 domains, the TIFI and the Jas domain. So we could show that basically Ninja interacts with the TIFI domain, that you can see here and here. So only a small piece of less than 50 amino acids. And the other way around, Ninja interacts to its C domain, which was another domain. And it interacts with the TIFI domain of Jas through this one. So you can see that here. So this piece is enough. Now, we still don't know how it becomes a bit bigger, but still not sufficient to really explain why this complex can repress recruitment of mediator and RNA domains. So we were lucky because proteomics improved, KTR improved the technology, so we could do TAP again. Sounds boring, but it made a big difference. So we do Jas, MYC2, CoI1, and now also Ninja. And I'm not going to step by step anymore. But basically, you see through the improvement of the method, for instance, that we, with CoI1, we no longer only pick up Jas proteins, but we also pick up the whole so-called SEF complex, for instance, in this case, the CoI1 and the ASC subunits, which are really essential for the stability and activity of CoI1. We didn't pick them up before, but now with the type of technology we do. And then you see all the same things happening, but there was a new player now visible, which is called Topless. This one I did not invent myself, it was there, the name already. And TPR stands for topless related proteins. So there are in Adopsis there are 4 of them, so we found all 4. And basically you see that they are there with the Jas Ninja complex in the absence of JA, but not in the presence of JA. Same for MYC2 because they block. And they're always there with Ninja, and you never find them with CoI1, just like the case for Ninja. We did need to hybrid again. We confirmed that Ninja interacts, show is in this slide. But I will start with this. So topless proteins were actually known co-repressor proteins. And why? Because they're pretty big proteins. I think they have thousands of amino acids. And they are there always as a tetramer, and then they recruit minimally 2 ear domain proteins. So you get a tetramer times 2 with all the rest attached to it. So usually you get mega Dalton complexes, and then you can imagine that this is really like a block of concrete that you put on your promoter on your protein so you cannot run. So that was exactly the case here. We found that Ninja has a so-called ear domain. It's a really simple domain. There's 5 amino acids. So if you find those, it's a very typical motif. You can show that if you mutate this ear domain, you basically have no longer interaction with topics. So now you really have a huge block that prevents the recruitment. And we actually measured the size, and with the Jas complex, we came to 1.5 megadaltons. It interacts with all this. So now we have a clear model. MYC2, in the absence of JA, is bound by Jas, and the Jas actually recruits this Ninja topless complex. When you have JA or stress, there basically JA is a leucine will connect the Jas with CoI1, just will be degraded. And basically, Ninja and Opus can no longer interact with MIC2, and you get your JA response. So, ultimate proof was, of course, to show that if you have a Ninja mutant, that it makes more metabolites and or it's more defensive, and that was indeed the case. We focused on the roots, and this came later, actually, this nice proof. So we had a collaboration with Anne Osborne, and Anne Osborne works on phytobeans and phytobeans produced specifically in Adoptis roots, and they are actually important for interaction with the microbiome and determine which they provide resistance, but also synergistic effects on beneficial microbes. So if you have a Ninja knockout, so you can see that this pathway is nicely inducible by JA. So these are all the intermediates of the pathway. These are all the genes involved, so you can see that you have nice activation by JA, and actually you have much more activation in the Ninja mutant, and you also have higher basal production of this compound. So indeed showing that if you remove the concrete block, you get constitutive metabolite production, which is not there in the wild type roots. Of course, remember the growth defense rate of Ninja plants are also smaller. They're not as small as the B2 overexpresses, but they're clearly not happy of fully developing either. So again, just knocking out Ninja will not give you good engineering. We thought of another tool. We tried that. Is again based on our histohybrid data. So we knew MIC2 can interact with the gases directly, and we also found out that this happens through an N-terminal domain of MIC2. So MIC2 has two domains, the N-terminal and the so-called BHLH domain, which stands for basic helix, loop helix. And this is actually the DNA binding domain. So these BHs typically act as dimers, and the dimers will bind the DNA through this BHLH. So it's also not unsurprising that the gases interact with another domain. Now, what was interesting to see is that there are actually 9 BHLHs known in the opposite genome that can bind the gas, and all of them had a pretty conserved aspartic acid residue at this particular position. So we actually made a mutant version of MIC2 where we replaced the D with an N, and then you can see that you lose the interaction with most of the gases now. So theoretically, you now have the MIC that would always be free. You expect that in Adopsis, and you can see qPCR. You can see that in both cases you have nice overexpression of MIC2, either the wild type or the mutant version. But actually, only in the case of the mutant version you get constitutive activation of the JA response. Plants look normal. We were happy. They were not small. So there we have our tool. But then we had germination rate super poor. So the better they were expressing, the lousier they were. We couldn't keep them for a year, and seeds were just dead after a year. So people still ask me for that line. I would say sorry, it's dead. They love it. So again, no tool. We're not there yet. We will get it. Okay. Then we realized it was we're still on top of the gap rate. We still have this one single, very general transcription factor. We still have nothing specific. It still activates all the metabolites and it still represses all its growth. So we thought we need more data and more insights. And then we realized that, well, those days you just do bulk transcriptomics. But from report lines, you can see that there is really also cell-specific expression within the organ. And you lose that if you just harvest your whole root of your whole CP. So, for instance, Ninja doesn't matter, it's expressed everywhere. But for instance, MIC2, you can see that in Adoptis root tips, it's really only expressed in the outer cilius, the epidermis cortex. And that, for instance, fits with the expression pattern of the phytobean genes I was talking about. So they're also in root tips, they have very nice epidermis cortex specific expression. So again, when we started realizing that there was a new technology that made my life easier, in this case even developed and installed by someone in the department, that's the label, and that's called single-cell RNA-seq. I'm not going to go really into the details, but just the basic. So for single-cell RNA-seq, you need to be able to separate your cells from a specific organism. So with Adoptis roots, it's relatively straightforward. You can protoplast them. It's very efficient. And basically what's also important, it leads to protoplasts from every single cell type. So if you protoplast some material, sometimes you lose this type of cells, and then you cannot do single cell because you lose really a piece of the organ. with arosis works very well. And basically, you get all your cell types protoplasted. Once you have that, you sort your cells and you go to a 10x genomics. So this is a commercial available item, which actually sorts your cell through a microfluidic system. And then you can do Illumina sequencing, and you get actually a transcriptome of each cell separately. Lots of heavy bioinformatics afterwards, and then you can start actually, thanks to the knowledge that we have on arosis, identify which cell belongs to which cell type. So you have then xylem, phloem, columella, endodermis, whatever type of cell type, and you can get these type of so-called UMAP plots, which shows the distribution. Now, you can also do this in mock NJA conditions, and that's what we did. So basically, we did the whole procedure by now mixing mock NJA treated roots. And what you can see here, this is mock, blue stands for the mock treated cells, JA for the orange treated cells. So you see that the overall shape is not changing. You just get a shift, which means that this was just a 6-hour treatment. It means that every cell is responding to JA, but it doesn't change the nature of the cells. So you don't get new cell types. You just get cells that start defending, but it doesn't change the nature of the cell. If you go into more detail, this is what you see here. You still have all the typical cell types. You just have a small switch from a defending cell to a growing cell. And then you can look at your pathway of interest. In this case, TUS is the marker gene for the strikapine biosynthesis. And what you can see here, so this is a violent plot. And I can briefly explain the principle. Every dot represents a cell. The higher the cell, the more it has the particular transcript. And then you compare to the left, you have the mock condition, to the right, the JA condition. If you have a JA responsive gene, you get an orange violent hub. If there would be a mock, the gene repressed by JA, you would get a blue violent hub. So here you can see, not black and white, but in general terms, you see activation and really specific expression of your diurnal pathway genes in the outer cell layers and very little to no in the inner cell layers. And that fits with the reported line. So now we have a tool. Remember, we have really 10,000 of transcripts. You can also see like which transcription factors are correlated or not with these target genes. So these three people, Alex, Jo, and Andres, started digging into this RNA-seq data, and then you get these used matrices. So what we did was to put all the known diurnal or tritopene biosynthesis in as a bait, and then we looked to what's co-regulated. We started with the top 100, and we were relatively lucky. So at the very bottom of the top 100, we had 2 transcription factors, again from the BHNH family, not so far away from MIG2, but not capable of binding JAS. So that makes sense because we were looking for something else. If you see the pattern here, you actually see a very nice match with the diurnal, which should be the case. Very little to nothing in the inner cell layers, and nicely JA responsive in the outer cell layers. So what we then typically do first to test out is use transactivation assays, in this case protoplasts of another plant, tobacco, where you overexpress your candidate transcription factors, and together with a reporter gene consisting of the promoter of this tritopene pathway gene, P5 phase, you compare that with most situations, and you see whether or not your transcription factor can transactivate your target gene. And that's, for instance, very nice in the case with MIG2, that was expected, but also we could confirm that these 2 major factors could indeed transactivate every single gene of the tritopene pathway. Next step is then to go to mutant plants to actually demonstrate that our genes are also active on Planta. This was more complicated because there are 3 lead factors and 4 of these mutations that we need to have septuple mutants. So Jo actually managed to do that. So we have both the triple MIC, the quadruple BHRH, and the septuple mutants. We group them, we treat them with Jasmonate, and then we look whether they produce more or less tritopenes. These are the genes. You can see here, I will just compare the first one with the last one. You can indeed see that this is blue MOC again, orange JA. You can see that you have repression or less activation and less basal expression of your pathway in the septuple mutants, and that corresponds with the accumulation of compounds. What you can also see is that's not gone away. So that means we're still missing transcription factors, We tried to find them, but we didn't, and we gave up. So we really downloaded the response, but it's not going there. So there must be some set of transcription factors which are bound by JAS that we don't know of and that we still have to find. Now, what we still didn't know or understand is why it's not there in the middle, because JA is perceived everywhere. So why it's still blocked? So, and I'm not going to give all the details. There are different possibilities, including chromatin condensation, but that's the case. So the only logical explanation of those days at that stage looked at either you need an additional set of co-activators, which are not regulated by JA, or a set of constitutive repressors, so in the outer layers, or a set of constitutive repressors in the inner cell, right? So we mind even more now, and now Klaas van der Poelen comes in the picture. Klaas van der Poelen in our department it's a mathematics person, has developed a so-called TF2 network, where you can actually now start integrating promoter remote persist motive data into your single cell RNA technique. So now you can start predicting which other transcription factors will potentially bind to your targets of interest, and then hopefully you get a cleanest. We tried it, and I was not hoping too much, but it turned out to be fantastic. So normally you get a mix of 30, 40, 50 transcription factors of all kinds of families, and you don't know what to do with it. In this case, we basically got a long list. That was not the case. That was not definitely solved. But there were only two types of families basically enriched. One was the so-called homodomain glabra family, and the other one was the DOF zinc transcription factor family. And I've put them here in blue and orange as a kind of a spoiler. So these turn out to be activators, and these indeed turn out to be repressors. And here you can see just two examples. We again used the protoplast system to show they were transactivating. Here you see the accumulation pattern. Most of them are very lowly expressed, but as you can see here, these co-activators are not expressed in the inner cell layers, but only in the outer cell layers, and they're not regulated by JA. If you use the transactivation, you also see that, so GL2 is one of them. This is again the TAS reporter gene. GL2 alone can not do anything. Tried all these homodomain effects, they don't do anything when you use them alone. Only if you combine them either with the BHLHs or with MIG2, you get a real synergistic effect. A real synergistic meaning that alone it does nothing, and it actually amplifies with several factors that take. You see the opposite picture with the Doppler factors. These are always there in the inner cell layers, hardly there in the outer cell layers. And again, you can see they're not repressed by JA either, so they will really always stay there and somehow block MIG2 acting on these target promoters. And they're really dominant, as you can see here. So if you, again, with the protoplast, let's say, you take MIG2 or the BHLHs, this would happen if you would have the BHLHs, for instance. If you add this DAG1 here, for instance, it really eliminates that with the BHLH. So really, very nice block. Now, we decided that we also made the mutants, and for instance, with this one, we see if you make a mutant, you have lower cytokine production. With this one, there was no effect. So we stopped there and we submitted the paper. Reviewers were a bit disappointed that we have no phenotype. Model was even very critical, but actually, I think it shows with this model how beautiful the system actually is. So the model that we find now is that JA is perceived by CoA1. In these outer cell layers, we get degradation of JAS, will activate MIG2. MIG2 will act directly on the tritopene genes, also indirectly by BHLHs. And it only happens to the outer cell layers because you have these co-activators there, only there, and you have these repressors sitting here constitutively. And actually, I showed you the list. These show the activity was similar for most of them, so many of them can repress, and actually shows that the plant really is making sure it doesn't have these metabolites in any other place than where it wants it to be. So most specialized metabolites plants are actually as toxic for the plant themselves as for any other living organism. So the way why plants actually are capable of making all these specialized metabolites is because they make them only in very dedicated environments where they can store them or release them. In this case, these triterpenes are there to interact with the microbiome. So when you have your root cap falling off, you get the triterpenes released in the medium, and that's where you want them, and nowhere else. And actually, the plant makes sure it cannot be somewhere else because it's really low level. So couple of slides to end. So I told you we're also working on translating this research into applications. I have 3 examples. I'm only going to talk about one. So basically, you can use this knowledge on transcription networks to engineer alcohols. I will talk about that. You can also engineer crop, food, or taste qualities. In this case, we use chicory. And we can also use that knowledge to study new molecules. If you start mixing genes from different plants and you put them on the JA control, you can make whatever you want. I'm not going to talk about that. I will stay within the theme of this talk, namely the transcription networks. And we use that in the plant Catharanthus roseus, or the Madagascar periwinkle, which is also growing here in Brazil. I have no idea why they call it Madagascar. It goes everywhere except in Belgium. So Catharanthus makes a lot of alkaloids, hundreds of different alkaloids, and there are 2 that are commercially very interesting, namely VIB-Active and Vincristine, very complex molecules, and they're very efficient anti-cancer drugs. Now, it's also super complex pathway. So, so far I've only showed pathways which are there in one cell type, in one organ. In Catharanthus, you have different situations according to the organ, leaves, flowers, roots, and within the organ even there is lots of transport. So the pathway involves more than 30 enzymes, all of them controlled by JA, except the last one here, which makes it complicated to engineer. And then there's lots of transport within the organ. So the first steps, which you see here, only 8, only take place in these so-called IPAP cells. The compound that you see here will then move to the epidermis, where there is another part of the pathway. This is a monotherapy, will be condensed with the derivative of tryptophan in the epidermis. You have a lot of additional steps. And then at a certain stage, here, these compounds will now move to specialized cells, which we only find in Catharanthus-related species, so-called idioblasts in the lattice first. And then you get the final steps, and you end up with your biomolecule that is stored in the latex of these plants, where again can do no harm to the plant. So very complex, but still everything is controlled by JA. So you can see that here. If you treat against cell culture or whatever organ of Catharanthus with JA, you get a very nice activation of every single enzyme except the last one in Catharanthus organs. You see everything conserved. The genes, the meat are there, and you can also see the transcriptome again. The amplification loop is there. The chances I didn't put that here, but also the other. At those days there was only 1 or 2 transcription factors. Now you also find them in the transcriptome part of the amplification loop. But this was not enough to be able to engineer it, because we only have 2 transcription factors for only a limited set of the target genes. So we used all the RNA-seq data that were available, and we did very heavy transcriptome analysis. And then you can see that you actually have very nice co-regulation of these pathway genes on a general level, but also within the subcellular level. So we combined JA treatment data, but also with subcellular transcriptome data. And then these 3 people, Alex, Maite, and Fadel, they developed a very efficient transformation system, because Catharanthus is very hard to work with, so you cannot make stable transformants those days. So they developed a flower infiltration system, which was really in today's measures both transcript effects and metabolite effects. And it was very high levels that you could get. And these flowers also make alkaloids, so they're good enough. And they have also all the same cell types as these, so it's a perfect model system. And then they mined all the transcriptome data, and they tried to match every single transcription factor to, for instance, the pathway in the IPAP cells, the pathway in the epidermis, and then the pathway in the idioblast and the lattice cells. And we actually were very enthusiastic, because we did find specific transcription seemingly correlated with every single cell type, and thought, okay, we'll now solve the whole thing. So they used this system, and here you see the color code. So the orange genes are regulated by Me2, the blue ones by this BHLH4A, the same one as in Argopsis. Only this first part is only 8 genes, and then ORCA, which is an AP2RF factor, regulates all the green genes. You can see this is unknown TF. There's lots of yellow stuff remaining, and we didn't manage. So we didn't find any additional regulator And this pathway is still not under our control. So that was a bit disappointing, but Fabian nonetheless continued with what we had. And he started engineering the flower battles, so the different tools we had available. And we actually started with this deal requesting tool, because now we're looking at the random system, so we don't care too much about those. And then you can see if you we used, I'll take this because for the color code. So we use either the Catalantus MIC2, the wild type version, or the mutant version, or the Arnoxis MIC2 wild type or mutant version. And then you can see that you only have transactivation of the target pathway genes with the mutated version and not with the wild type version. And it's also reflected on the metabolite level. But as you can see here, there was not many effects on the downstream alkaloids. You only have, back to the pathway, you only get up here. And maybe some of these, which makes sense because we only work with two, so everything downstream is not going to be activated. So not good enough. So Fabian continued, and then he started combining everything we had. And I will just show the ones that worked. So at the end, focus on this one, the light green, no, sorry, this one, the purple blue one. Igor is better in color code than me. Cyan blue. So that's the combination of this one, which is BHLH, with the de-repressed MIC2 and one of the ORCAN AP2 RFs. And then you can really see that now we activate many more genes, including transporters. So these are 3 transporters to guarantee the communication between the cell types, and also many genes. And you see here the activation level, it's 1,000-fold for some genes. So we really have a very specific now activation of the pathway. And that was also reflected on the metabolite level. So we still see some increase in strictosine, but now it's really translated sometimes 10,000-fold increases in the alkaloids and flower methods. Unfortunately, we did not get to the industry in the VIB because we miss these non-J irregular genes. So there is some feedback on the whole pathway, but not until the very end. So that's still beyond our control. But at least we're very close, and you can really engineer titrate out all of the jazz. And that sometimes happens in some tissues. Sometimes it's severe consequences. There is also other splicing for the target genes, but what also seems to be important is chromatin looping. So we're also looking into that. So MIG2, the box that's recognized by MIG2 is basically almost present in every single gene in the genome. So there must be more that drives the specificity of MIG2 to determine which target gene. And it seems very complex, but there's also a difference between long distance or long-term signaling and short-term signaling. There are short-term effects of MIG2 and long-term effects of MIG2. That's regulated by chromatin. So there's definitely a lot of other processes that drive response. There's also transgeneration memory, again regulated by epigenetic effects. So there's much more. We started with that because we had to find out which were the players, but now we have to open up, and that's for instance why we go to other technologies like the 2.D, where you can actually see in which environment MIG2 is sitting. That's kind of and the affinity purification was not for MIG2. I didn't stress it, but as you could see with the top, we only had repressors there. So the only thing we put down with top was MIG2 in an inactive state. We never had mediator, we never had the article, and that was strange. Then we found out that during the procedure, you actually have to make a native protein extract, and you lose the protein bound to the DNA. So you resolve information of this activity. So you need other methods. But yeah, and then indeed we need to go on. And then we're also looking now at E3 ligases that regulate the stability of the transcription. There are tons of E3 ligases activated by JA signaling and regulate levels of organ. Because I didn't mention, but many of these target genes that are reduced 100-fold transcription level and increase 0 on the protein level. And we don't know what's happening there. It's very strictly regulated protein homeostasis. So you may wonder why you need 100 times more transcript to the protein is not moving. And then we found one E3 ligase once, we took it out, and then suddenly the protein was going up, and that E3 ligase was co-regulated to JAs. So there are definitely many more systems. And again, because of the ecological importance, you really need to keep JA response under control or your plant stops growing. So everything we found actually basically is things that repress the pathway. And once you unleash it, it's gone. And then you get new plants that don't grow anymore.
I have a few questions. First, I noticed that mediator 25 is one of it's like physically interacting with all
MIG2.
With MIG2, okay. Because I know that in the lignin field, they found out that mediator 5, it's controlling actually the phenylpropanoate homeostasis.
Yes.
And once you have like a loss of function, normally you have like a lignin mutant, then you have a growth penalty.
Yes.
So when they cross this lignin mutant with the mutant of mediator, you recover the growth. So somehow it's like controlling the growth as well. Yeah, exactly. So is there any correlation on a way that you can, in a way, because we had already another talk in our course here, talking about this trade-off between growth and defense. And the guy was trying to actually uncouple things, like just to produce a plant that is actively defending and still growing very nicely. Exactly. Everybody's trying that. And is it like loss of function of this mediator in the Jasmonate? Can you like have increased Jasmonate responses? And maybe the growth is also
loss of function. You have less med 25. You have less of Jasmonate response mutant, for instance. It also has oxen effects because also the arcs from the oxen response they also interpret them. Right? So you get there's a very close interaction between the oxen and JA pathway at different levels. But I think that the work from the Med5 and phenolics may be actually regulated by the metabolites. So what we sometimes see is when you overexpress a single transcription factor, to take the example from Catharanthus, we overexpress this VHLH that only targets the first 8 genes. You see the whole pathway responding, which means, and we do the transcriptome, and we for sure know because we test all the promoters that these pieces are not acting on those ones. So the plant senses when a piece of the pathway is activated, and I think there are other enzymes and or transcription factors that are bound by those intermediates or end products, and then either repress part if there's too much, or stimulate it when there's a piece missing. Because again, these intermediates are typically very toxic, so you don't want them. So there's all kinds of feedback again, and that that's why another technology that we've developed is LibMS to find protein metabolite interactions, because we want to see if there's some kind of sensing of metabolites. And going back to the example you mentioned, I think it's direct, potentially, putatively, there may be lignin, intermediates that may bind to mediate. For instance, there are also terpenoids known to be bound by topless. And then you get direct feedback from your end products on topless. It's not really super hard data, but it looks convincing enough to believe that there's metabolites of all kinds can impinge on transcriptional machineries, even if you don't have a real transcription expression. That's what we're also looking for, but that complicates the system. So many of these metabolites may bind either to enzymes or regulators and get additional things which are not controlled by JA directly. But again, make sure that the plant has it's always the most important thing is to have your pathway complete. Once you start taking pieces out, you run into danger, also the plant itself, of course. That's also why you have this typically in the system. It's also the principle you apply. You look for co-regulation. It's not because to make your life easy as a researcher. It's because the plant wants to make sure that everything happens at the same time. And if you compare that, for instance, with other kingdoms like bacteria and fungi, what they have is physically clustered pathways. So every single gene of a pathway for one metabolite in the cell it's in one piece of the genome, and they only need chromatin condensation to regulate the expression of the pathway. Plants have water because typically all of the genes are just scattered around the genome, so they need something else, and that's why they have this co-regulation system.
That's why prokaryotes are much more smart and they have the overalls.
And also about the experimental design of your single cell experiments. You're showing one of your interaction data that you need 1 minute to see something, and then something, and you said it was 6 hours treatment. Yes. So how did you decide this?
Very good point. That's over the years. Because sometimes we miss things that that should happen. So we know that ubiquination, there were papers from the human field, happens in milliseconds. Oh, yeah, but we cannot work at it. So the task to do is 1 minute. But you always, again, it's about balances and quorums. And if you look at the ecological picture, the plant should not immediately start, because for instance, JA is also important for shade avoidance. So if the cloud passes by, you don't need shade avoidance. So you have to really build up, and you really have, I think you need a certain threshold before the system becomes really activated. Then you have to decide what you want to match. If you want to see it, you pick the ideal moment. In this case, also with the top 1 minute for that, an hour for another protein, transcriptomics. Depending on the process, we go for 15 minutes or 24 hours. Again, that changes. And to make it, one of the things we're puzzled about is that you see the transcripts activated within half an hour, typically, for metabolite pathway genes. But the metabolites, we typically only detect after 24 hours, 48 hours. And it's not that we below the threshold, because there is a basal level of most of the metabolites that we've got. But somehow, maybe it has to do with protein homeostasis, somehow we have to wait 24 to 48 hours before we actually see the metabolite going up. But sometimes the transcripts don't need to be done at that moment. And then, because people in the past said, you have to integrate. So, but I was stuck because I had my transcripts at half an hour until 6 hours, they went down, and then my metabolite was coming 2 days later. I was stuck because I would never have any correlation with my transcripts and metabolites. It was totally useless. I can't explain it, but it's what happens.
Splicing, alternative splicing, and a lot of things that we are missing. I mean, that's obviously we are really reductive.
Lots of things to discover.
I suggest to go. I don't know if we have questions.
I think the audio was not working for some time. So.
I have a couple of questions. You mentioned, talking about the Cataranthus, you mentioned that you are running Nick, 2YB with Nick.
Yeah.
Are you working with roots?
But not the Cataranthus, eh? In Tiroli, we only do in tomato.
Okay. I thought that you, okay.
No, Cataratus is a very complicated model system. So that's why we start working with the flowers. And we try to do lots of things that now is better because now there's a good genome in the single cell in Cataratus. So what I've shown here, we could actually revisit. And sometimes I put things in the later. So now what we know, we could actually revisit and see if we do find, but it's much more complex than we thought because I was naive in the beginning and I thought, oh, I just need one additional regulator or activator for my cell-specific expression pattern. From what I've shown in Aropsis, you see it's super complex. Yeah? We have there 20 genes that you don't see with co-regulation. They're just at the constitutive level. And they're as important as the modulated ones.
And how much redundant are the 3 nicks?
Totally. So the only difference in terms of activity,
because I mean, you showed a TIA,
and
the effect is not the same.
TIA is the same. The only difference is their expression pattern in the plant. So that determines the specificity. But if you would take MIG3, you put it in the same cells as MIG2, it will do exactly the same. So they interact
but
physically.
there is an organ specific.
Yes.
Okay.
There's one question on YouTube asking, thanking you for the excellent lecture, and asking whether the regulation of GA signaling is also concerned in monocots.
Yes, but it's more complex in monocots at first sight. So there are many more receptors. So most plant species only have one CoI1, except the monocots. I think wheat, for instance, has 7, corn has 3.
But that's because it's with hexaploid.
Yeah, but corn has 3. So yeah, it's true, but
With corn.
but that doesn't only explain it because you have tobacco only has one CoI1 as well. It's tetraploid. So no, there are much more JASs and much more CoI1s. Mix, I don't know, but for sure the receptor, the co-receptor complex is heavily amplified. We don't know why or purpose. There is, to some extent, organ and cell-specific expression, but it seems to be very important for instance, in heat tolerance and monocots. But people are only starting now. So in rice, there's a lot of activity with JA signaling, and they try to disentangle different CoI1, and rice is one of the easiest monocot model systems there. But it seems to be there an additional layer of complexity. At the same time, monocots have lost some JAS subfamilies, so there's clearly some evolution. But the JA compound is again the same as in the dicots. And MIG2 is also there, and probably more specific other specific, but it's there, but more elaborated.
These 3 terpenes that you showed, you said that they are produced more like in the outer layers of the roots.
In the root tip.
In the root tip. So they are produced upon elicitation, let's say, or they are like stored as glycosylated forms because they're like specific environments. I mean, or both maybe. I mean, it's something like that is triggered to be produced, or they are also stored.
Both, but they're not stored as glycosylated versions. Somehow they just can stay in the cells. But you also with the cell death, the cell death they get excreted. And you have acetylation, which makes them slightly more soluble, so they can be excreted in the direct environment. I didn't show that here, but if you go up in the root, they go to the inside, and you lose part of the pathway. You get other terpenes coming in, so there are more functions, and we're studying that right now. And actually, MIG2 also goes inside. You go in the differentiations of the so it's more complex than we thought. So again, depending on the specific role of your compounds in that particular organ, in this case the root, where you have exudation, and it seems to be important for the effect of the microbiome. There is a lipid outer cell layers. If you go up, it goes inside, and it's connected to vasculature. But we don't know what.
Well, Well, that's great because, for example, some nematodes,
Yes.
Biologia incognita, they go into
Yeah.
the roots through the root tip, and then they go to the middle
Yeah.
towards the phloem.
Yeah,
So it makes completely sense for
we did nematode assays, but there's no difference in the phenotype, unfortunately. With our losses, which is not the perfect host for nematodes. We tried, but we also thought that there must be something. We also don't know if could be a way of transporting up the phytoplasts to the leaves, because the pathway is really only there on the roots, nothing in the leaves. But maybe it goes up. Nicotine is the same. All the people know nicotine is in the leaves. But if you look for the genes involved in nicotine biosynthesis, they're only in the roots and only in one specific cell type of the root and nowhere else. And all the nicotine is immediately transported.
So more questions? I have to say that I'm disappointed that you didn't present the work with Título, the Belgian project.
I had some slides in the past, but I think I ran out of time.
So that's it. There's any more questions? So thank you, Alain, for your contribution, and thank you all for your attendance.